For Lecture 2, we're beginning with a discussion regarding other killing mechanisms that our white blood cells use that are not phagocytic. These are kind of interesting. For example, eosinophils, which I mentioned are the red staining cells, they do secrete toxins. I also mentioned that part of their role is to kill the larger parasites. So a parasite in your body might get surrounded with these eosinophils, and the eosinophils will dump their contents onto the surface of the parasite to attempt to kill it. Therefore, if you have really high levels of eosinophils, which is called eosinophilia, you might have a parasitic infection. Although eosinophilia is also associated with people that have a lot of allergies as well. A way to trigger eosinophil activity is if endotoxin, the molecule we talked about during the virulence factor discussion, the LPS found on the gram-negative outer membrane, is present. That is a trigger for immune responses. Our innate system gets activated. One thing that happens when LPS is present is that cells, like eosinophils, will dump their mitochondrial DNA out into the surrounding area, and it will make a web-like barrier to trap and block organisms so they can't go very far. This is a suicidal event for the cell, but one that is a last-ditch effort to contain a situation that's perceived by the cell to be dangerous for the organism. Something similar happens with neutrophils, but I'll come back to that in a minute. NK cells are natural killer cells. Their job is to secrete toxin onto cells that are not self. They're interesting because they're considered not specific, even though they're lymphocytes, which are a part of the specific response. So they exist as a bridge between innate and acquired immunity. They recognize cells that are not self, and they will try to kill those cells. Coming back to neutrophils, if you recall, those are the cells that are first responders. They're phagocytic cells, meaning they kill by phagocytosis, but they also do some other important activities too. Interestingly, important to their killing abilities is oxidative killing. Their enzymes generate superoxide, a toxic oxygen radical that does damage, and together with their destructive enzymes, kills the cells they ingest. The other ability neutrophils have is that they form these nets, which stands for neutrophil extracellular traps. And literally, it's a cell suicide mechanism to contain infection. So remember, the neutrophils are first responders. They come on the scene to clean up, and as they get overwhelmed, they will initiate a suicide pathway where they dump all of their contents out. This in turn ends up making a big web of DNA and proteins to trap and immobilize stuff. So when the macrophages, the big guns, come in, they can clean up the mess. It's really interesting. This slide lists two different types of receptors that are under our non-specific chemical defense. They do respond to a limited range of signals. Over evolutionary time, our bodies and other organisms have learned to respond to things that are pathogen-like, and they're called pathogen-associated molecular patterns, PAMPs. These are molecules from cell walls, flagella, LPS, those sorts of substances that are common to many different bacteria. These toll-like receptors, or TLRs, are on the surface of cells. You can see a diagram right here. There's a variety of these toll-like receptors, and they bind these PAMPs. And that activates a pathway inside the cells so you can initiate inflammation and apoptosis, which is cell suicide. They also will get the adaptive immune response primed and ready to respond. There are now known to be 10 toll-like receptors. The name comes from Drosophila flies. There was a receptor called toll that had been studied in Drosophila, and then scientists studying mammalian immune response noticed that a gene for a cell surface receptor looked a lot like the toll receptor in the flies, but it was in humans, hence the name toll-like receptor. Toll-like receptors are extracellular. They're on the surface, as diagrammed up here with the arrow. They detect substances external to the cell. But NOD proteins, they're intracellular. They're inside cells, but they also recognize PAMPs. So if there's viral proteins or pathogen components that are inside, like bacterial cell walls for intracellular pathogens, then NODs are going to recognize and trigger a cellular event inside the cell. Often, the response is something that initiates transcription to turn on genes, but they can also cause the cell to undergo apoptosis, cell death, cell suicide. The NOD proteins can also stimulate an inflammatory response in that surrounding area. So while these responses are not specific, they do recognize patterns that are indicative of organisms that are not self, right? In other words, invaders. The next thing that we're going to talk about is the part of the second line of defense that is nonspecific, the interferons. These molecules are made in response to viruses in general. Basically, the type 1 interferon is antiviral, and that is what we're going to be spending time going over. Type 2 is phagocyte activating, but we're not going to focus on this. Interferon type 1. Here's the process. What happens is that a cell might become infected with the virus, and the cell is going to send signals to neighboring cells to warn and alert them that the virus is around and to prepare themselves for possible viral infection. This figure from your book is explaining this process. In this first panel at the top left is a cell infected with the virus, and that's going to stimulate that cell to release interferon. Interferon is indicated by this IFN right there, and you can see it's being released to the neighboring cell. The neighboring cell is going to pick up and recognize that signal. Meanwhile, the cell that was first infected might die, but the neighbor that received the interferon signal, it's going to make antiviral proteins. These antiviral proteins will not really do anything yet. They're not active at the moment. However, if that cell that was infected releases virions and the viruses now infects that prepared cell, the inactive antiviral peptides, the AVPs, become activated. Once activated, they're going to play a role in degrading messenger RNA and will bind to ribosomes with the purpose of stopping or halting protein synthesis. Now, what does that mean? Well, if you can't have proteins being made, you're not going to make more viruses, right? Will this cell die? It might. But if it dies, it's because it is not able to resume protein synthesis. It also means that the virus is not going to continue to replicate and spread. So this cell might be sacrificed so the rest of the neighboring cells can be saved. So the purpose of interferon is to prevent cells from replicating virus and continuing that cycle of cell infection. Make sure you understand this process of making interferon and what the interferon is doing for the neighboring cells. The last thing we're going to talk about in this section is complement. I mentioned in the previous lecture that complement is a series of proteins and it's found in the liquid portion, the serum of the blood. The role of complement is to break apart or lyse foreign cells. It can be activated in several pathways, which we'll talk about in a minute. But besides lysing cells, it can do a couple of other things. It can increase the ability for macrophages to phagocytose cells, and it will also increase inflammation. Now, the product that's responsible for lysing cells is the membrane attack complex, MAC, and it's the last substance formed in the activation pathway. Now, complement is activated by one of three pathways: the classical, alternate, or lectin. Those are the three activation pathways. I don't want you to remember all of the things that are in between, but I want you to know, one, what is the activating pathway? Two, what the result is, which is the membrane attack complex. And three, the sidebar over here, what other things can complement do? There are three ways to activate complement. All three of the pathways, although the initiation is different, once activated, they all converge to this little box in the middle. You do not have to remember what these different proteins are doing, but they ultimately assemble a membrane attack complex. If you look at the figure over here, the membrane attack complex looks kind of like a little barrel or a pore. It's going to be an opening that lets everything leak across the membrane. You can see a real transmission electron micrograph here of a MAC complex. So a cell that is riddled with these MAC complexes will basically leak out and it's going to die. That is the purpose of complement, is to kill the cells. But at the same time, once it's activated, it will also increase inflammation and it will facilitate the activity of a phagocyte to carry on phagocytosis of something. That's called opsonization. Opsonization is coating a substance in a way that increases phagocytosis. What are the different pathways? Well, the classical pathway is activated when antibodies recognize and bind something. This is a bridge between the adaptive and the innate immunity because it involves antibodies, which come from the adaptive immunity, the topic of the next chapter. The alternate pathway is activated with things like endotoxin, aka LPS, and various glycoproteins that are very bacterial-like. Then finally, the last pathway is the lectin pathway. Lectins are proteins that bind carbohydrates. If you look here, this cell, probably a bacterial cell, has mannose on the surface. Our cells do not have this carbohydrate. So if mannose is detected, lectins bind to that and flag it as something that is not recognized. That will activate complement. So when lectins bind, that will activate complement. Those three are the start of the activation of complement. They all converge on a pathway that ends up making the membrane attack complex. Make sure you understand that. The very last topic is inflammation. But really, the only thing I want to highlight with inflammation before I stop is that inflammation is typically a good thing. If it's acute, it helps activate things so that you can heal. But if it's chronic, it is typically bad. Inflammation involves redness, tissues get warm, you get swelling and pain. But what is going on is that your blood vessels, your capillaries, are dilating to let more blood flow through the area, which leads to the redness. The capillaries themselves increase in permeability, so fluid is going to leak out. This leads to swelling, and your white blood cells are stimulated to migrate out of the capillaries in the area, and altogether, that is going to initiate repair of tissues. So acute inflammation is actually part of the healing process. If you have long-lasting chronic inflammation, it is not a good thing. There's a video tutor that's in Mastering Micro that you might want to look at that goes over inflammation. This image is reviewing margination and diapedesis occurring in the environment of an inflammatory response. And this figure is the figure going through inflammation, acute inflammation from your book. Oh, and yes, one more final concept before we leave innate immunity. The ability to spike a fever is a response to attempt to slow pathogens' rate of multiplying. Pyrogens are substances that trigger our hypothalamus to raise our body temperature. LPS is a known pyrogen, but other substances can be too. So fever is part of innate immunity. The ability to respond is already present. It's not a learned response. So we're going to stop here. This is going to be the end of our overview of non-specific immunity, your innate immune response, covered in Chapter 15. Up next is specific or adaptive immune responses.